Hi. I am going to this semester try to do some advanced recordings, which are going to be preparation. So the idea will be to just introduce some core concepts, which you can read more about in the textbook. I've posted the sections that are relevant, but I think that maybe having these to summarize key terms might be really helpful. I may not have time to do them for every single lecture topic, but I will certainly try, especially at the beginning, because that's when people are really sort of grappling with a lot of new terms. So for this first one, I am going to cover materials from lecture 2, which will be Friday. And I will talk briefly about regulative versus mosaic development. These are just models for ways of thinking about development. You will find through the semester that most types of embryonic development are a combination of both concepts, but they were devised early in the study of embryology, and so they're a little bit restrictive. We'll talk very briefly about induction. I'll spend more time in the lecture talking about the different kinds of experimental techniques that have given rise to ideas of induction and what induction means, and organization of tissues and so on. And then a chunk of time on the role of genes in development. It seems to me from my most recent teaching of the topic, which was last summer, that there is a need for a little bit more information just about how genes work, especially at the beginning, as you're starting to get used to the topic. Okay, so that's already taken a bit longer than I expected, but hopefully these videos are not going to be more than 15 minutes long. I don't want them to be too onerous, but I want them to be helpful. So please ignore my messy artistic abilities. I want to start with mosaic versus regulative development. These are, as I said, two different models. And historically, it was thought that most invertebrates develop through mosaic development. And the idea of that is you have a fertilized egg. It is a single cell. But if you look inside the cell, you will find that different areas inside contain different quote-unquote determinants. So we'll talk more later in the semester, because of course these determinants actually exist, and so we will talk a little bit about what they actually are and how they work and so on. But the idea was, if you look at the fertilized egg, there's a part of that egg that's going to form the back. We call this the dorsal, and there's a part that's going to form the belly of the organism, ventral. And then there's a part that's going to form the head. This isn't true for all organisms. Some have different kinds of symmetry, but we'll use this. And so this would be the anterior part, and then the tail is the posterior part. So these are terms you're going to hear more about later on, but the idea is then that if you were to look inside the egg, there would be a bunch of determinants that were specific for dorsal, the back, and posterior. So we would call that the DP cell, just as an example, or the DP region. And then the belly would be ventral, and then the back end would be posterior, and then anterior, ventral, the belly side, but the anterior end where the head is, and then dorsal and anterior. Okay, and this isn't, I mean, as I said, not all organisms are exactly divided this way. There are some that have different kinds of symmetry, but if you thought of just like, a fish or a reptile or something, you would see sort of head versus tail, back versus belly. And the idea is that there are different determinants. They weren't completely sure what those were, because of course they didn't know about DNA at that time, or much about protein at all. But the idea is that you've got determinants, and that they are found in this region, so that when our cell actually then divides one into 2 cells and then into 4 cells, we now have 4 cells that contain the determinants for just certain kinds of cells. So in this case, this cell now has the physical ability to form dorsal structures on the back, but just at the posterior of the organism. So that would be, maybe muscles and things for the back end, etc. Think about most organisms, the anterior versus the posterior, the structures are different, the internal organs are different, and so on. And this other cell here contains the information or the ability to form just anterior structures, but on the back side, and the cell ventral structures at the front, and so on. And so the idea is that now each of these cells has a restricted cell fate. We'll talk more about cell fate later in one of the other introductory lectures. But the idea is that now what you've got is restricted cell fate. So I would like you to think in advance of the actual lecture. So suppose we were to do one of the earliest kinds of manipulations that were done in embryology was to remove a cell and see what would happen. So if you could somehow just remove this cell and then leave the embryo as is, how do you think development would proceed? What kind of observations would you expect to make? And then this was, as I said, a historical model, but so what this is most related to and the kinds of things to be watching for in upcoming lectures is the idea of maternal factors that are placed in the egg by the female and are therefore present in the embryo in specific places inside the egg that will then direct a particular type of development. And hopefully when we go through the lecture, you'll see some examples that give you a better idea of how this works. Regulative development was different. It was historically thought to be how vertebrates develop, but there are examples of invertebrates that are that allowed for the idea of regulative development to be developed. That definitely not only vertebrates. And the idea is that the egg is it has the potential everywhere to form everything. And then the development proceeds and it depends on the kinds of interactions that occur between cells, different gene regulation, and so on. And it's flexible. So the idea is, your cell divides and you now have two cells, one of which might be like what we would call dorsal, and one of which might be called ventral or anterior posterior, or however you like to think of that. But that and then those signal to each other and send information back and forth, and over time as the cells divide and you have a more and more complex embryo, different cells kind of know where they are based on the interactions that happen between them and the cells nearby. So it's regulative, and the reason why it's called regulative is that it can respond to environmental changes. It can respond, of course, to the signals and information received from other parts of the same embryo, but also experimental manipulations. And as you can imagine, that is what gave people the idea that regulation was possible. And so if you were able to separate these two cells, and you have a cell that's supposed to only form dorsal structures, for example, but if you do the experiment and you find that, actually somehow the cells are able to reorganize and divide and still produce the organism, we would call that regulative. And then sort of contrast that with what would happen if development was entirely mosaic. Okay, so as I said, those were two models for thinking about how development takes place. And you will find as we go through the different organisms and how their development proceeds is that there will be aspects of both of these in pretty much all the types of development that we observe. And I think, I'll talk about induction, and then I think I might have to do a second video just for this one. So I want to talk a little bit about induction. It's something you're going to see examples of throughout the whole semester, so you want to watch for that. And it's a case where a cell or a group of cells will actually influence the development of a cell or cells nearby. So this is what we would call a close range interaction. And the kind of responses are highly variable. So either the shape or the behavior, I should have had a comma here. And by behavior, we mean things like maybe the cells start migrating or moving around, or maybe they start sending out processes to make connections, like if it's a nervous cell or something like that. So the cells do something in response. They might change their rate of mitosis, so they might start dividing more quickly, or they might slow down the rate of mitosis. And then, of course, among all of this is changes in cell fate. So the idea is you've got this group of cells and they're sitting there, but when they receive information and signaling from nearby cells, they are induced to alter their fate and then maybe become part of the nervous system or part of the muscle system or something like that. So there are two components. There are the inducing cells, the ones that are sending the information. And when I say sending, sometimes the cells are physically touching each other. So it's not like they're sending long distance. These are close range type of interactions. And then you have the responding cells. So the inducing cells are sometimes also, if it's a whole tissue, it might be called an organizing center, which we'll see an example of in the lecture. I will talk about early experiments that actually showed that some tissues act as organizing centers and therefore as inducers, and then the responding cells. And something to pay attention to is that the responding cells, it can't just be any cell that's nearby. They have to be what we call competent. They have to be able to respond to the signals that are being sent by the inducers. That means that they have the right proteins that can receive the signal, and the right proteins active inside the cell that can go ahead and change the cell's shape, the behavior, or the gene expression in response. And we will go, as I said, this is just to give you an idea of what we're going to be talking about, and I will go through an example, and I'll ask you some questions on it as well in the lecture that will help you understand what this is referring to. I will spend a second video just going specifically over genetics background. If you took 202 recently or 302 or some other course where you understand these questions, what a gene is, what gene expression means, and what phenotype and genotype are, there is no need for you to go through the second video. If you're feeling a bit rusty or unsure about those terms and what they actually mean, then you might find the next video helpful.